All right, ladies and gents, welcome back. So today we're going to talk about how the neuron actually is capable of sending a signal. And from AP standpoint, this is really more their focus. You need to understand the anatomy in order to understand the physiology, but ultimately AP is really looking for you to understand function above and beyond everything else. Um, I will say this can sometimes be a bit of a difficult concept. There's a lot of information here, and understanding all of the little pieces and how they connect is really important. So don't be afraid to come back and rewatch these a few times. Spend your time in the textbook, spend your time in the activities to really ensure that you understand how this works. But what we're going to talk about today and over the next few videos really is about the cell's membrane potential. So when we look at a neuron, we're looking at this voltage across your plasma membrane. And really what that means is a difference in electrical charges. How do those electrical charge differences occur in cells? Well, it's going to come back to its chemistry. Really, it's ionic chemistry and how much of the concentration of one ion versus another on opposite sides of the membrane is going to allow us to set up that difference in electric charge. Today we're going to focus really on just how is this charge established in a neuron that is not sending signals. So what we're really talking about today is its resting potential. It's going to ultimately be the change away from that resting potential then that causes your neurons to actually transmit and process information. So we're looking at changes in those differences in voltage. That's what's then going to allow us to elicit a signal response. So first off, as soon as a neuron sends a signal, we have to return it back to its baseline or form this resting potential. So how does it happen in our cells? So what we look at in a mammalian neuron that is at a resting potential, we're really dealing with the concentration of two ions, your sodium and your potassium. This might sound familiar. We've talked about sodium potassium pumps before, and they are highly important in cells like your neurons. Why? Because they're going to be what allows us to set up the resting potentials and then eventually the signaling of information down those axons. So when we look at a neuron at rest, we look at a neuron that's not sending any signal. Their potassium ion concentrations are highest inside your cells, while the sodium ions are highest outside. Now, that doesn't mean that they are the only ions that are being taken into account. If you take a look at the picture, there's things like chloride. There are also other miscellaneous ions as well. But we're going to focus mostly on the sodium and potassium ones for our discussion here. And it really is because that sodium potassium pump is going to be huge in reverting these concentrations back to normal once that signal gets sent. So, in order to allow this to occur, not only do you have your sodium potassium pump, which is what you're looking at right here. Remember, we load sodiums from the inside, pump them out. We take the potassiums, load them from the outside in. You've also got ion channels here as well. Okay. And the opening of those ion channels also allows for the conversion of that chemical potential energy, that change in electrical potential across that plasma membrane. So your neurons, when they're at their resting potential, right, not sending a signal, they've got lots and lots of potassium channels widely open so that the potassium is flowing to the outside of the cell, and we're using the sodium potassium pump to bring them back in. On the flip side to that, there are not a lot of sodium channels open. So we're constantly pumping sodium out to bring potassium in, but we're not letting a whole lot of sodium diffusion occur, bringing that sodium back in. So what this does is it results in a resting buildup of negative charges inside your cells with an overall positive charge on the outside. So if we look, here are your inner versus exterior cellular concentrations, right? Potassium, much higher on the inside, much lower on the outside. Sodium is its opposite. Then you've got things like your chloride with that negative charge, not a whole lot on the inside, much more on the outside. And then you've got other large anions, things like charged proteins, peptide chains. Again, relatively high on the inside, but not really so much on the outside of your cells. So if I take this collectively, that's where that positive overall net charge ends up on the outside, and their overall net charge ends up on the inside. So negative inside, positive outside, when that cell is not sending a signal. So how do we know this? Well, we can actually model resting potentials in a lab setup. So your text talks a little bit about this. You've got these artificial membranes that will separate two channels, and we'll put a voltage meter in the either side of that membrane. Concentration of things like sodium chloride are much higher inside your cells. So what does that do? We put that in an aqueous solution. Remember, ionic compounds dissociate. So you've got your K plus and your Cl minus. If we put a much lower concentration of KCl on the opposite side, well, what do your ions want to do? They want to go from high to low. So if we look at this, that potassium diffuses down its concentration gradient if we allow for this open channel to occur. However, you end up with an overwhelmingly larger buildup of chloride ions inside your cells, right? So potassium chloride comes apart, potassiums want to move to where they're lower, and we've given them the ability to get through. However, I put that low concentration of KCl over here. The chlorides are hanging out. There's a couple of them, but I have not provided a way for that chloride to get across the membrane. So overwhelmingly, that chloride gets built up in here. That's where that negative charge comes from. And because I'm allowing the potassiums to get to this side, I get that overall positive charge on the other side. So what we're looking at is this equilibrium potential, and I've given you this equation, this Nernst equation. The video that I'm going to show you in just a minute talks a little bit about this. Take a look at it. It's kind of interesting information. That being said, don't get lost in it. It's not something that's going to be a major portion of assessment. But for those of you guys who maybe take some sort of anat and phys courses in college or other higher level classes in college, you may come across this. So I figured it's not a waste of your time to just give you a little bit of exposure. So what is this Nernst equation? Its job is to take a look at the particular ion of interest, whether it's sodium, potassium, chloride, or something else, and be able to talk about the membrane voltage that it creates from that particular ion if we're assuming the cell is in fact at equilibrium, meaning it's not sending any sort of signals. So when I look at the equilibrium potentials, so I look at those values for potassium versus chloride, that's where those positive and negative values come into play. So in a resting neuron, the currents of sodium and potassium are equal but opposite. Okay? And so that resting potential across that membrane remains steady. So what's the big takeaway here? Your membrane potentials can change from its resting value when the membrane's permeability to a particular ion changes. So if I leave the permeability to sodium, potassium, chloride, any other ion sort of consistent and steady, you will get that overarching equilibrium to be met. And so the cell can't send any signal. But if something occurs, whether it's opening channels, using pumps, whatever the case might be, to change the permeability of that membrane to a particular ion, then I can change that voltage potential, and that's where I have the potential then to start sending signals, start changing those voltage concentrations and be able to do something with it.
Hey there, hey. I'm Allie Astrocyte, and you're watching Neurotransmissions. We're going to start talking about how the brain uses neurons to send signals throughout your body. You'll find that it's a lot like a giant squishy battery. And, like a battery, this process is electric. week, we found out that neurons are the information superhighway for sending signals from cell to cell, transmitting information across your whole body. But how does your brain actually do it? To make it a little easier to visualize, let's look at a battery for a second. Keep in mind, this isn't a perfect analogy, but it's a good place to start. As you can see, batteries have three main parts: a cathode or positive end, an anode or negative end, and an electrolyte, a solution between the two. Chemical reactions within the battery cause electrons to build up at the negative end. These electrons are unstable and don't like to be packed up with their fellow electrons. Like wild horses or an ex-boyfriend, they need their space. This means that they will try to move to a place with fewer electrons whenever possible. When a wire connects the positive end of the battery to the negative end, this lets the electrons run down the wire. Usually there's something along that wire that uses those electrons for power, like a light bulb lighting up. The electrons are trying to reach an equilibrium, where they're as far apart from each other as possible. Once the equilibrium has been reached, the battery is used up and is dead. It has no more electrons left to flow, and so no more current. Your brain uses a similar process, except it uses something your body is jam-packed with: ions. If you've taken a chemistry course, you might remember that ions are charged atoms and can be positively or negatively charged depending on if it gains or loses electrons. Need more information about atoms and ions? Check out some of these other YouTube videos. In the nervous system, the main ions are sodium, potassium, and chloride. Let's take a closer look at how those ions send signals. Here's a neuron. All cells have a membrane called the phospholipid bilayer, which is very good at insulating the inside of the cell from the outside. Built into this membrane are channels that allow ions to move easily across the membrane. Imagine that this neuron has a lot of positively charged ions inside of it, mostly potassium, and some negatively charged ions, like chloride. Since the inside of the neuron has a very high concentration of potassium compared to the outside, the potassium ions diffuse down the concentration gradient. That is, they try to even out their concentration between the inside and the outside of the cell, just like the electrons trying to move down the wire on the battery. This means that the potassium ions flow out of the cell through the channels, leaving behind their negatively charged friends. These negative ions build up a negative charge inside of the neuron. Eventually, this negative charge starts pulling on those outward moving potassium ions. We end up with a balance between the diffusion forces pulling the potassium out and the negative charge pulling the potassium back into the cell. This is called the equilibrium potential. This creates a potential difference, or a voltage, just like a battery, across the membrane. But just like a battery without the wire, the positive and negative ions build up along the membrane with nowhere to go. When the cell isn't sending a signal, this is called the resting potential. Most neurons have a resting potential of about -70 millivolts. Not a whole lot, especially when you consider that a AA battery has a voltage of about 1.5 volts or 1500 millivolts. Resting potential can be a very complex idea to explain, but just so you know, even this is a very simplified version of resting potential. It's really much more complicated, mostly because the concentrations of other ions, like sodium and calcium, have an effect on the electrical potential. There are all sorts of calculations used to measure it, such as this equation, called the Nernst equation. We won't cover it in a short video like this. But if you would like to learn more, check out the description for more information. For now, let's focus on some things you should remember. Potassium is much more highly concentrated inside the cell, and sodium and calcium are much more highly concentrated outside the cell. This will be important going forward. So, now we've got this neuron with a negative charge inside the cell and a positive charge outside the cell. How does this transmit a signal? Where's the action? Patience. Like I said, this is a pretty complex concept. Come back next week and I'll tell you all about the action. The action potential, that is. I'm Allie Astrocyte, and until our next transmission, over and out.